With us now are occupational health nurse Chris Pontus, who is Associate Director of Health and Safety with the Massachusetts Nurses Association, and Dr. Kevin Kavanagh, who's the founder of Health Watch USA. So thank you both for being here. Go to Cap with question number 2.
Sure. So Dr. Kavanagh, this question's for you. My understanding is that mRNA is a pretty new technology that's being used to create some of the COVID vaccines. First, can you help me to break this down into its component parts? Like, what is a virus and what is mRNA?
Well, yes. A virus is really quite a simple organism. In fact, some people don't even feel it's alive. It is a capsule. In this case, it's a round type of a capsule that's composed of phospholipids. There are little spike projections on that capsule, which are called the spike protein, that attaches the virus to the cell and helps it enter the cell. And inside this capsule is mRNA, or RNA in this case. It's an RNA virus. There are also DNA viruses, but COVID-19 is an RNA virus. And that is all it is. And when this RNA is injected by the virus into the cell, it acts similar to mRNA, or messenger RNA, and it codes the cellular machinery in order to make more viruses. And that's all it does. And then the viruses get made in the thousands and it spreads. It cannot reproduce by itself. It needs a host cell. There is no organs, no tissue, nothing within a virus but a capsule and the genetic code for producing another virus. This is it, and it will take over the cellular machinery. It does not enter the DNA or the nucleus of the cell. And so that is kind of the basis of a vaccine, because your body mounts an immune response to the virus, and it amounts it to the entire virus. But what is most beneficial is the immune response to the spike protein, because this is what the virus needs to attach to the cell and to function. And so antibodies to that spike protein are key. And so the vaccines are designed to make a very specific immunological response to that spike protein. And how they do this is by injecting the RNA code, or the mRNA, for just that spike protein. Not the whole virus, just the spike protein. It gets injected into your arm. It doesn't go anywhere else in the body, just in the arm. It will enter a few of your arm cells, and from there, those cells make not a virus, just a spike protein. And that creates an immunological response composed of neutralizing antibodies, very specific antibodies, which will then kill future viruses. And so that's the idea behind an mRNA vaccine. And we really have 3 types of mRNA vaccines. 2 of them are produced by Pfizer BioNTech. The second is then produced by Moderna. These are pure RNA vaccines. They are encased in a phospholipid capsule, which is made in the lab. The mRNA is made in the lab. It's printed out by a machine. There's no natural virus in that, and that's what's injected into the arm. Now, Johnson & Johnson does this a little bit differently. They have a DNA vaccine. In other words, they have just the DNA for that spike protein that they put within an adenovirus capsule. That infects the arm. The adenovirus capsule causes a better immunological response. It injects the DNA into the arm. The DNA then codes mRNA, and this is all in the cytoplasm of the cell. It doesn't enter into the nucleus of the cell or affects the genes whatsoever. This is all outside the cell. The DNA injected makes the mRNA, which then makes the spike protein. So Johnson & Johnson vaccine has an extra step. Why do they have the extra step? Because DNA is more stable. It can be stored in the refrigerator. It doesn't require this ultra cold storage, this reconstitution. And if any of that in transportation or in bringing the vaccine up to body type of a temperature, if any of that is done incorrectly, the vaccine can lose its effectiveness. So the Johnson & Johnson is much easier to handle, easier to store, and easier to distribute. But they all work on the same theory of getting mRNA to make the spike protein. So that, in a nutshell, is what we're looking at with a vaccine, mRNA, what it is, and what viruses are. They're kind of pesky little things that you would think wouldn't give you any problem whatsoever. They're not even alive. They can't even reproduce without the help of a cell. And the last thing I'd like to add is that mRNA is extremely unstable. That's why you have to do the ultra-cold storage. So it degrades and disintegrates immediately. So it's not like it sticks around in your arm for a very long time. It enters the cell, does its thing, and it's gone. So these are safe vaccines theoretically, and as we've seen in the last 6 months, they look like they're very safe in our population.
Well, thank you for that breakdown. How long have scientists been researching mRNA for use in vaccines?
Well, the short answer is since 2011. So that's been quite a while. But they were originally made for vaccines to give immunity to cancer cells. And they then adapted these to the coronavirus. So they've been around a long time. They've been studied a long time. They've been used in people, cancer patients, for a long period of time. And so they're really not new. It's not a brand new technology. But as far as using them for infectious disease, this is, I believe, the first time this technology has been applied for that purpose.
So similarly, even though COVID-19 is new and deadly, other coronaviruses exist, right? And how long have those been studied?
Well, definitely. The first major coronavirus was SARS, and that had a pandemic or a large epidemic between the years of 2002 and 2004. After that, there was MERS. MERS was a very fatal, very severe infection that caused outbreaks and also a pandemic between the years of 2012 to 2015. With MERS, 3 or 4 out of every 10 patients died. It was very fatal. And interestingly, when an epidemic is highly fatal like that, and people are severely sick, and you can identify who's sick, it's easier to contain, and it usually dies out. And that's one of the problems that we have containing this COVID-19 pandemic, is that we have all these asymptomatic people. The majority of people don't get severely sick. It's hard to identify cases, and people are spreading it all over. So the genetic code of coronaviruses have been studied for years. And this is the virus which people and scientists have thought is the most likely virus to cause a severe pandemic on a global scale. So this was expected in many countries. Many countries were highly prepared for this. The United States, unfortunately, was not, which is unfortunate, and we are suffering the consequences at this point. Now, I should add that the common cold is also a coronavirus, but that's a different type of coronavirus. So we're not talking about that type of coronavirus.
So Dr. Kavanagh, one of the major concerns that I've heard from a lot of members is that they think the vaccine was rushed to production, that these things happened too fast. This coronavirus just came on the scene about a year ago. So how do we have this vaccine so quickly? Can you tell me about how clinical trials for vaccines are typically run?
Well, yes. Vaccine production has in the past taken many, many years. And this has been based upon taking a virus out in the wild, modifying the virus so it's safe to inject into humans, and then a prolonged type of testing and evaluation. One of the big differences here is that we've known about this coronavirus for some time, so that there was a big lead time in vaccine development. In addition, with the Moderna and Pfizer vaccines, these are printed out like on a printer. I mean, they're man-made. When they have to make a new one or modify it, they just insert the new code and print out a new virus, and they're good to go with testing. So it's entirely different than trying to harvest something from nature that's unsafe, make it safe into a vaccine. No, these are printed from the get-go. But let's talk about the timeline, and let me talk a little bit about clinical trials. There are usually 3 phases of clinical trials. There's phase 1, which involves about 10 to 100 healthy volunteers, and its goal is just to determine overall vaccine safety and that it doesn't cause really severe side effects. And then there's a phase 2 trial, which is looking at safety and efficacy, and that's usually between 100 to 1,000 subjects. And again, it's a small trial. It mainly is looking at safety because with that small a number of people, you're maybe not going to pick up efficacy. In other words, you have to have a fair amount of people infected to compare two groups. That's too small of an N to do that with. And finally, there's phase 3, which is anywhere between 1,000 to 10,000 individuals. And the goal there is safety and efficacy. Overall, these vaccines have gone through at a very rapid pace. The preclinical stage was already done. Moderna had a vaccine ready to test within weeks. Again, they just had to print it out. That was the technology that they had. Phase 1 was extremely fast, and phase 2 and phase 3 were essentially combined. A lot of these were overlapped because they had a large number of volunteers. So a lot of these phases were run concurrently. So where you were looking at 32 months, sometimes the phases were cut down to a few months. And phase 3 is the most controversial because during that phase, you like to have at least 6 months of data. These vaccines had an emergency use authorization by the FDA after 2 months of data. Now, the justification for that is that the vast majority of vaccine types of complications occur within the first 2 months. Not all, but the vast majority. Now, everybody in that trial that reported data at 2 months, which was in November, they're still in the trial. They're still collecting data. So right now, there is 6 months worth of data, and data will be collected for a total of 2 years on these individuals. And everybody that's received a vaccine, there are a number of data collection mechanisms to survey for complications and also for efficacy. Right now, the vaccines appear to be highly efficacious with the wild variant of virus, but they also at the same time have very low incidence of serious complications. In the United States, there have been over 100 million vaccine doses administered, and there have been no deaths which have been attributed to the vaccine. This is unlike 1,000 to 4,000 people dying a day of COVID-19. So overall, these are very, very safe vaccines. And if you want to know the size of the trials, Pfizer-BioNTech had more than 40,000 people enrolled in their trial, Moderna, 30,000 people, and Johnson & Johnson, 40,000 people. So overall, there were 120,000 individuals which have been tested by a vaccine which uses mRNA to code the spike protein and elicit an immunological response. And our data right now is 6 months as far as efficacy and safety. So we're in a very good position to say that these are safe and efficacious, and let's get the vaccine.
That's all really good to hear. So, Dr. Kavanagh, in your opinion, when thinking about the amount of work and research that has gone into coronaviruses, COVID-19, vaccines, and the clinical trials, yes or no, were these vaccines rushed? Can we trust them?
You can trust them. They were expedited, but not rushed to affect safety. And you have to realize the 2 or 3 years of production was crunched down. They started producing these vaccines at risk, hoping that they will work so that they would have a production line in factories ready to go. So a lot of things were compressed at great expense to get these vaccines out and delivered to the population. So the answer is, can you trust them? Yes. They are much safer than taking your chances with COVID, and especially safer than taking your chances with COVID variants.
So how should we compare the risk of vaccine side effects to the risks of staying unvaccinated?
This is going to be a long answer because we're going to go over a little bit of risks of COVID. We're not going to concentrate on the deaths because the younger people, well, they're not too concerned about that. They're immortal. COVID doesn't affect them. That's what they think. So let's talk about medical problems with the overall realization that survival is really a pretty low bar of existence. Okay? There is a lot more to health than I've just survived or I'm alive. And there are a number of long-term medical problems from COVID. First of all, there is a huge problem that we're just starting to understand regarding the long hauler syndrome. This can affect people with mild or moderate infections. You have to remember this virus affects every organ of the body, so that a number of people will have different symptoms. It's very difficult to have one particular category and saying, this is it. No, this virus affects a number of different organs and everybody presents a little bit differently. If you are hospitalized, you have about a 20% chance of having heart damage. This includes either a heart attack from the blood clotting or actual infection of the heart itself from the virus, and you can get a myocarditis. And this can come on and produce symptoms many years down the line. But studies have shown a very high percentage, approximately 20%, of people who are hospitalized have heart disease. Even higher percentage, up to 80%, have reported having difficulty breathing at up to 9 months out. Greater than 9 months? Who knows? They've just studied it for 9 months. And these people are still having trouble breathing. If you look at people that are hospitalized, 29% have to be readmitted and 12% will die. And if you take all the COVID-19 patients, mild, moderate, and take a look at how they're functioning mentally, they have a much higher incidence of psychosis, depression, seizures, anxiety, brain fog, and this has been reported in between 20 to 70% of patients after COVID-19. And this includes even young individuals. At 6 months out, there was a study. Now, this could be even more than 6 months. The study just stopped at 6 months. 63% of hospitalized patients still complained of muscle weakness and fatigue. 25% had difficulty walking. They couldn't walk as far as they thought. And 22 to 56% had problems or impairment with lung diffusion. Again, that's trouble breathing. So these are really chronic problems. And a new article, which just came out this week in JAMA Network Open, and this studied 9 months out, 30% of those having COVID-19 still had symptoms. And they evaluated 177 patients who had COVID-19. 85% were not hospitalized, which means they had either mild illness or moderate illness, and they were still reporting symptoms. 49 of these 177 patients, they reported having at least one persistent symptom in follow-up. So that is disturbing. 9 months out, they're still having problems. And if you look at hospitalized patients, it's even worse. 80% trouble breathing, greater than 80% having a cough, greater than 80% feeling feverish. So the take-home message is, just because you survived COVID-19 doesn't mean you're going to not have problems. And just because you're young does not mean that you're not going to have problems. You need to get vaccinated. Now, all of that data is with the wild type or original virus. And so let's talk a little bit about the variants, because the variants are more infectious, they're meaner, and they cause more severe illness. The United States was first infected with COVID-19 with the Wuhan variant. Well, within a couple of weeks to months, we had a new variant called the D614G, and this was felt to have originated in Europe and came to the United States. It was more infectious. People weren't sure if it was more fatal or if it caused more severe disease, but that's what we dealt with over most of 2019, was this D614G variant. Then, in the later part of 2019, we started to see the UK variant, first in England and then over in the United States. And this is the B117 variant, more infectious and it is also more virulent. It has an increased risk of causing severe disease. Luckily, all of those type of variants, which let's call them generation 1 of the virus, they are dealt with very well with these vaccines, and prior infection would be expected to give you fairly good immunity to reinfections. But then we had another group of variants which is currently infecting the United States. And these variants have two different types of escape mutations. The first one is called the EEK variant. I guess it's spelled EEEK, but it is the E484K mutation, and it's an escape mutation on that spike protein, and that is in the New York, South African, and Brazilian variant. Prior infection has been reported not to give immunity to these variants. Dr. Fauci testified that in the Johnson & Johnson study out of South Africa, reinfections occurred, and it appeared that no immunity was given to the South African variant from prior infections. And so that is very disturbing. The same has been noted in Brazil, where they've had huge surges. Cities in the Amazon that had 76% of their population that had been infected or immune to the wild type were then just devastated again with the Brazilian variant, that too having this EEK type of a spike protein. And then we have another variant called the California variant, homegrown in the United States, which has an additional mutation, which also makes it more infectious and more virulent. And scariest of all is the double mutation in India, which has just this week been reported in San Francisco. And this has two of these escape mutations on a single virus, and no one knows what that effect is going to be. So here's the problem. If you're vaccinated to the wild type, is it going to help with the escape mutation variants? And the answer is yes, if you get both doses. It will blunt your illness. These vaccines are up to 6 times less effective with these new variants. But the difference between the first dose and the second dose in antibody production is tenfold. So you've heard the first dose you get immune, second dose tenfold. That's enough to help you with these new variants, but it requires both vaccinations. So you say, well, what about Johnson & Johnson vaccine? That only has 1 dose. Well, Johnson & Johnson is the only vaccine that's been tested in South Africa. And that is a huge advantage because they actually have noted that that vaccine causes a significant reduction in severity of illness. And it has efficacy of approximately 60% against that variant. Now, Johnson & Johnson, you say, is not as good as the other vaccines. Well, it was tested against later types of viruses that were more virulent and harder to take care of. So that is the one vaccine that was actually field tested. The other vaccines, we just have laboratory data to say, yeah, antibody levels look like it'll work. Johnson & Johnson was tested in people. So let's go ahead and get this vaccine and get both doses. It's very important because of these variants. So as you can see, you have a choice between death, long hauler syndrome, which occurs quite frequently, and that's if you get COVID-19, probably be even worse with these variants. If you've had COVID-19 before, don't count on it giving you any protection to the variants, at least these new stage 2 variants. And if you get the vaccine, you will have at least good protection to the wild type, and you're greatly increasing your chances of having a less severe illness to these new, more malignant variants. And as I said before, over 100 million doses of vaccine have been given out in the United States. No one has died yet from the vaccine. And on top of that, these vaccines now have 6 months of safety data and counting. They keep collecting data as the months go by. So we are very lucky. I would like to tell you that I think that this is a miracle and proves how great science is. I just think that we were really lucky and we actually stumbled upon something that works, and let's take advantage of it, because if not, this pandemic will continue to spread. And by the way, the virus will continue to mutate. It mutates by spreading the virus. So, we need to cut down on the spread, and we also need to stay safe.
And the last question that I have is my co-hosts here, Ryan and Kat. They've both been vaccinated. I'm the only one, I think, on the show today who hasn't been vaccinated yet. So if I have a vaccine preference in mind, should I be shopping around to wait until I get the one that I want? What do you think I should do?
Absolutely not. Get whichever one you can get. Because here's the thing, the Moderna and Pfizer vaccine are very similar. Moderna is a little bit easier to reconstitute and store, doesn't require quite the cold temperature that the Pfizer-BioNTech vaccine does, but overall they're similar technology. The one difference is the J&J vaccine. It's single dose. It does have the advantage of being field tested against the variants, at least the South African variant, and that's a big advantage. And so if you have that vaccine, you may want to get that. And I should add that when you talk about the South African and the Brazilian variant, the exact same mutation is in the New York variant now, which is causing severe infections in New York and in Massachusetts. So you can take the vaccines that you think have a better number in efficacy, and I just mean the actual number, because that number was from the wild type virus when it was studied. It wasn't during today when you have all these variants. Or you can take the Johnson & Johnson vaccine, which is less efficacious on paper, but has actually been studied with the South African variant. And that vaccine prevented severe complications and deaths in all of their patients after 1 month of being vaccinated. So, it's a hard choice. So I would just get whichever one you can, because I think that they are both very good types of vaccines. And the only exception would be is if you're horrified and very scared of needles. Johnson & Johnson's just a single needle. The other two require 2 shots. And of course, if you ask my grandchild what she would want when you presented it like that, no doubt she would go for the Johnson & Johnson.
Well, thank you both so much for being with us. We really appreciate all the information that you shared today.
Okay, thank you very much.